Welcome everyone to another MetaCell Presents webinar. I am Giovanni from MetaCell, and I have the pleasure of introducing Sharon Crook and Rick Gherkin today from Arizona State University. They're going to talk to us about validating computational models, which is of course a very important topic, putting the spotlight on enforcing scientific method when developing novel computational models. So I met Sharon and Rick many years ago going to their talks, and a lot of you in the audience may know them already in the same way, but for those of you who don't, Sharon and Rick run the ICON Lab at ASU and are prominent members of the computational neuroscience community. They are behind projects such as NeuroML DB, the NeuroML initiative in general, and SciDash. So before we dive into the material, I want to spend a few words talking about MetaCell, and we're a software company that specializes in building neuroscience software. We do both simulation and neuroscience data visualization. We work with academia, not-for-profit, and industry partners. You can see some of our partners here. As you can see, we have the ASU logo as well. And as we have an ongoing collaboration with Rick and Sharon here with us today, translating some of their ideas into software, and we might see some of that in the material today.
Thank you, and thank you everybody who's there listening and watching, and thanks again to MetaCell for inviting us to do this webinar. It's great. We're happy to be able to have this opportunity to talk to you today about validating computational models. So we're moving beyond some of the stuff that we're involved in around reproducibility, and thinking beyond that to evaluating all these models that are now becoming available to our community, and evaluating our own models in a more formal way. So some of you might have seen this this quote from George Box, that all models are wrong, but some are useful. And I think it's important for us to keep this in mind as we create our models. And when we think about how do we determine which models are useful, we're really looking at the question of trying to formalize how we think about our models, how we think about what it is that we're doing when we constrain them, what kind of data are we using to constrain our models, and what kind of tests are we really using when we say that our model is a good model. A lot of times, if you go to the literature and you look at a model that's available, associated with a publication, it's not at all clear what the evaluation criteria were for a particular model that's being shared. And so I'm going to be mostly talking about models that we use in neuroscience. And so I think it's illustrative to think about some of the many different kinds of models that we have in neuroscience, right? So from conductance-based models that are typically very data-driven to reduced models that still might be data-driven but focused more on the behavior of a cell rather than the mechanisms that create it. Of course, there are other kinds of models like statistical models, models for machine learning. They're also driven by data. And so for different kinds of models, the way that we want to evaluate the model might be a little bit different. All right, so here's another illustration of this idea of evaluating models and how we might think about comparing models to experimental data. So everybody's familiar with ideas from cosmology and how our models of how the cosmos works have changed over time as more and more data have become available. And so you might think about formalizing those ideas where you think about different historical models, you think about observations that were made over time, and early models explain some of the first observations that were obtained. And so we might think about saying that those models pass, but when we compare those models to more recent experimental observations, they don't explain the observations. Whereas more complete models that we might think of as being better models, explain all the data that are available. And so this is the idea behind validation of models that we'll be talking about today, is this idea of formalizing how we think about models, how we think about experimental data, and being very clear about how we compare models to data, and about how we share that information. All right, compounding this problem is the fact that there's more and more experimental data available over time. So this is looking at how many articles there are in PubMed over time, and you can see how huge the number of articles are today. And so it makes it hard for us to harder for us to be clear about exactly which data, which results a particular model might be expected to agree with. All right, so the question is, how do we evaluate models against all the relevant data for a particular model, where we have to be able to say what those relevant data are? How can we do it in a way that's more automatic? And how can we do it in a way that's public and transparent, so that it's quite clear what a model what data a model is meant to reproduce, and how we're evaluating how good it is. And so the answer to this, of course, is this resource coming out of our group. Rick is really the head on this project, but it's called SciDash, and the idea is to share collections of empirically informed unit tests, and then to be very clear about how we're evaluating models by saying which tests they pass. And so this idea really comes from basic ideas from software development, where when you're developing software, you want to know, you want to do this in a way so that as you're developing code, it's very clear what the code is doing, how well it's doing it, and then as you develop, as you make further developments to the code, you want to make sure that unit tests that are used during development continue to, that your new code continues to pass, your code continues to pass unit tests as you add additional functionality to the code. All right, so before we think about how we might apply this to models, I think it's useful to think about what models typically are. If we want to think about models in a very general way, we might think about the fact that a lot of models for neurons are really like a function that has an input and an output. And so more specifically, we might think about a model neuron as having an input that corresponds to how you're perturbing that model during a simulation experiment. So for instance, you might be injecting current, or you might use spike times to drive synapses, or maybe that model is embedded in a network that drives those synapses. But if you want to look at the behavior of a model neuron, then you need to be able to say something about its input. And then likewise, when we talk about the output, what we're looking at are simulation results that we can use to predict observations, right? So this is one way of thinking about a model. And even more specifically, let's consider a model for maybe something like a cortical pyramidal cell, like the one that you see here on the right. And let's consider that we are injecting current to the soma for some period of time at some amplitude. And then the output of the model would be a predicted waveform, a resulting simulation waveform that predicts how the membrane potential would act during this type of experimental result. And so typically, if we're creating this type of data-driven model, what we really want to do is compare our output to recorded membrane potentials from experiments on real cells. And so this is the idea of unit testing for models, is specifying very clearly, exactly what your model is, exactly what the simulation experiment is, and then being able to specify what type of, so what data you're comparing to, and exactly what is the sort of analysis routine that you're using to compare the simulation results to recorded data. So that's the idea. All right, so SciDash is set up so that there is a framework that's written in Python for handling this idea of unit testing for models. SciUnit is sort of this Python framework, and it's actually domain agnostic. So it can be applied to models of anything that you're interested in, and basically any kind of model, as long as it fits into this idea of having some sort of output that can be evaluated to either qualitatively or quantitatively to determine whether you have a good model or not. And of course, that evaluation is going to depend on the context. What did you create the model to do? You would be the one to decide how you wanted to evaluate, what kinds of tests would be needed to evaluate a particular kind of model. And then Neuron Unit is a library for testing models in neuroscience. So the kinds of unit tests that are there now are tests for looking at models of neurons, models of networks, and models at the subcellular level of things like ion channels, and comparing them to experimental data. But you could imagine that there could be tests that are more qualitative, that look at something like say a neuron's behavior. Does it burst? Does it have adaptation, or something like that, where a test might simply be something like, does it pass or does it fail the test? The test result might be something like that. All right, and then in addition, an additional part of SciDash is the SciDash portal, which is a website for sharing information about testing models. So, for instance, if you go to this website, you might see something about a particular model, a particular test, and then a score, either a quantitative score or a pass-fail score for whether that model passed a particular test. And Rick will talk more about sort of the details of the portal later. All right, so a nice thing about this validation framework, I think, is that it fits in well to a whole ecosystem of tools that are focused on reproducibility and rigor for models in computational neuroscience. And so, for instance, you might think about the FAIR initiative, findable, accessible, interoperable, reproducible for data, which applies to models as well. And so this fits in very well for that framework. For instance, these unit tests that are available in Neuron Unit are meant to be quite generalizable, right? So they're interoperable, they're focused on different data types, for instance, and so presumably you could reuse these tests in multiple different modeling settings. And I think this idea of evaluating models based on the tests that they pass fits in very well to ideas about making modeling more rigorous. So, for example, when you do publish a modeling paper, if you can state very clearly which data sets your models are meant to be compared to, and exactly which tests should be used to evaluate those models, then that leads to better transparency in our field. All right, so you could share these model evaluations at the SciDash portal, make a URL available. This is the goal, make a URL available that links these models and data and tests so that they're public, and this idea about how you constrained or evaluated your models and how good they are would be very transparent. All right, so there are many different steps that are necessary in order to make this framework work, from getting experimental data and preparing it to be compared to model output, finding models, figuring out what kinds of tests they can take, and then many steps that are needed for describing tests and actually using them. And so I'm going to hand things over to Joe for another poll, and then Rick will tell us more about these pieces of this Neuron Unit framework, testing framework.
All right, thanks, Sharon. So we're going to just do another quick poll to check if people are paying attention, but also to ask what kind of computational models do people in the audience normally work with. You can pick one. You just basically pick the most representative for you. So I just started the poll and waiting for people for votes to come in. Right, see votes coming in. And I'm gonna give it another 10 seconds. Right, see most of the audience put their vote in. Just another 5 seconds, and it looks like multi-scale models are winning. But let's share the results. So I'm just gonna go ahead and close the poll. And I'm gonna share the results with everybody. So it looks like most people in the audience are working with multi-scale models, but there's a good chunk of people also working with detailed single cells, and then everything else trailing after that. So, okay, so I'm gonna go ahead and add this and pass it back to Rick, who's already sharing his screen, I see.
Okay,
Right.
I am. Great. So yeah, so there's thanks Sharon for introducing that, and Joe for organizing this. So you can see here there's really I actually have no idea where this is coming from.
Yeah, that's back on.
Okay, I was not actually in slideshow mode, so I'm not sure.
Oh, so I think it's still sharing my screen now.
Okay, right.
So
Okay, so let's. Okay, so now Rick, you have control now.
Okay.
Okay. You see my correct screen?
Yes.
Great.
Perfect.
So there's really two major challenges to making this work. So what Sharon's described hopefully sounds very interesting to you, but also might sound a little bit like magic. So how can this be possible? How can you actually test all the models? And so there's two really big challenges. One is that, look, there's a lot of steps, there's a lot of things that have to be done, and they have to be done right. And people historically have done them in very different ways. So you look at any one paper, and someone might have gone through all of these steps in some way, but they might have chosen their own way. In another paper, someone may have done another way. So all models and all sort of informal tests would have been done using different criteria and different approaches. And so 10 years ago, something like testing large numbers of models would have been impossible. But what's changed since then is that a lot of these areas here have been there have been standards developed to handle each of these areas. So I'm going to give some examples here. There's just some examples. There's not just one standard often in the field, but sometimes many, and that can be okay. But here's some examples of standards associated with each of these areas that have to be handled if you want to be able to do model validation. And as with the way the standards help is that if you want to develop tools to do model validation, is having to support the way that each person, each scientist or each investigator made their models or the way they collected their data or whatever. You just have to support a few standards in each of these steps, and then you'll be able to tackle maybe half or more of the modeling ecosystem in a field. So, for example, if you just think about the experimental data format, neurodata without borders. Before, the experimental data in neurophysiology was sort of a wild west of ad hoc file formats. But now many large institutions and prominent labs are using neurodata without borders to share their data. So that really makes the extracting experimental data information to use to parameterize a test a lot easier. But I'm going to highlight just two examples here from this ecosystem. One is from data summaries that you can use to parameterize tests of already analyzed data, and the other is a place to locate models instead of having to sort of hand code them all from a bunch of papers that you looked up. So Neuroelectro is one. This is a project that I was involved in developing several years ago with Sridhar Tripathi, who now has a lab in Toronto. And the basic idea here was, okay, we don't want to have to go through and look at all the data for all the properties of every neuron, right? There's tens or hundreds of neurons, and there's been tens of thousands of experiments have been conducted upon them, and these results have mostly been reported in journal articles. What if you just go through and take the data from those journal articles and extract it with text mining techniques and put that into a database, and then people could say pick a cell type that they're interested in, and they could find all the data about that cell type that's been published. So that's what that project goal was, and it was actually quite successful. Here are just examples of 9 different cell types that are in the database, and you can see there's been data extracted from over 100 articles about the CA1 pyramidal cell with over 1,000 individual electrophysiology values that you could use to say what are the properties of this cell, what are the experimental properties, and you could build a model around that. So what you can see here is there's actually 235 different cell types that are carried in this database that have been reported from the literature, which is covering pretty much most of the brain. Some cell types not as well covered than others. You see some of the more popular cells here that people like to record from. But not only can you go to the database and look at this data, but there's actually an application programming interface that allows you to use a software like SciUnit to extract this data automatically without going to the website and use that to build a test. So in one line, you can build a test that says that tests the properties of a CA1 pyramidal cell, specific property, like say the width of the action potentials, or the resting potential, or the distribution of interspike intervals, or something like that, and use that to test a model of the same cell type. Another resource is the NeuroML Database. So this is something that Sharon has been developing with Justice Bergiolis. And here, it's a place to find models, but not just to find models, to find a very specific format of models. So NeuroML is a markup language, kind of like HTML, that's used to describe models. It can describe a wide range of multi-scale models from ion channels all the way up through networks. And when a model is expressed in NeuroML, you can do a lot of really cool things with that model that you couldn't do if, say, you'd hand-coded it in MATLAB. It opens up a whole tool chain of model simulation and introspection and now, of course, validation, and also as well as many tools for visualizing and computing many interesting things about the model. So if you, for example, go and search for a pyramidal cell in this database, you'll get several results. This is just a selection of them, of models. This isn't all of the models that people have ever made of pyramidal cells. It's just a few examples. But in all of these examples and more that are on the website, you will get, if you click on one of the links, a screen that gives you more information about the model, for example, where it came from. You can explore that model using other tools for modeling or model exploration, for example, Opensource Brain. You can look at the model on GitHub. You can also get the NeuroML files, and so the NeuroML files will allow you to sort of enter into this ecosystem of tools for model exploration and simulation. So another thing you can do with that is you can explore tests or at least simulations that we've already run on all of these models. So for over 2,000 models in this database, we've conducted a series of experiments, simulation experiments on those models, a standard set, and you can see the responses of those neuron models to all of these to this standard set of experiments. So this standard set of experiments actually matches the standard set of experiments that have been done to cells that have been released in the Allen Institute cell types database, and of course on Allen Institute's models as well. But these are sort of models that didn't just come from the Allen Institute. These are models that came from the larger neuroscience community. So we you can run these experiments yourself, or you can get the results that we've already done produced here, or you can get tests that have been parameterized using these kinds of results. The other thing you can do is you can look at the morphology of these cell models, and here some of these measures are the same ones that are reported using a tool called LMeasure, and they're reported on neuromorpho.org, which is a resource for finding full reconstructions of neurons. You can find tens of thousands of cell reconstructions there. So here are digital reconstructions of model cells, where these are not model parameters. Width and number of bifurcations are not model parameters. Those are computed from the model file. So because it's NeuroML, there's a tool chain that you can use to go through and compute a large number of things about the model, physiological properties and anatomical properties. And here's some of the anatomical ones. And you can go and you can compare these and say, okay, do these model parameters match the corresponding parameters in a reconstructed biological neuron of the same type that's found at, for example, neuromorpho.org. So going back here, there's clearly a lot of things to be done, but standards do help us solve the problem. And so now we get to the lower section of this figure here. There's still a lot of things to do with aligning tests and models. So once you have a test that's been parameterized with experimental data, and you have a model that's been instantiated from some model code, what exactly do you do to line them up, to make the model take the test? And so that's where these tools come in. So InsideUnit is sort of the core tool here. It's this Python framework, as Sharon mentioned, and it's domain agnostic, and it basically looks like this. If you wanted to see what the code looks like, it's from a user perspective, it's about this simple. There's a lot going on under the hood that I won't talk about today, but you can learn more about it later at the website. Basically, there are model classes. The model class might be something like a Hodgkin-Huxley type model, or it might be something larger like some sort of a circuit model, or it might be a reduced neuron model. And the parameters to that that get passed in would be like a model file that describes parameters of the model, or maybe some custom parameters that you've made up. And the test is sort of similar. There's a generic kind of test, like a test of the width of an action potential, or the distribution of interspike intervals, or something about the morphology of the cell. And the parameters of that are data that comes from a resource like neuroelectro.org, neuromorpho.org, or data you collected in the lab. And then the last part is judging the test, or judging the model with the test. And you get a score out, and that score is how well the model and the data agree. So advantage of this approach, this SciUnit, it applies to any scientific domain. So if you come to this webinar and you're not a neuroscientist, or you don't care about these kinds of neuron models, you still would SciUnit would be something you might be interested in, because you can write your own tests and apply them to your models. This makes no assumptions about model implementation. So this is really the key. This is really the second key to making all this work, along with utilizing community standards. You separate implementation from interface. That's sort of an adage in computer science. What that means is the test doesn't actually need to know anything about how the model is implemented. All the test has to do is use a common interface for doing things to the model, like running simulations, for example. And all the model has to do is expose methods that implement those things the test needs to do, like, for example, inject current into a cell. So if you have a model and you have a way of injecting current into the cell, that's something that you would need to expose. Or if you're using NeuroML, you don't have to do any of those things, because all that's taken care of for you. It also has rich test score visualizations, which I'm going to show in a little bit, that'll show you how well the models are performing on the tests. Very well integrated with Jupyter. So I've used Jupyter notebooks, which have exploded onto the scene in the last few years in data science and science in general for analysis. It's a great way to do your testing. And there's also command line tools. So for example, if you are doing modeling, and you every time you write a model or make a change to a model, you make a commit to GitHub, for example, and you want to see, just get a report. How did your test turn out? Or you want to get an alert if your test starts failing. That's all available through these command line tools. Neuron Unit is the part that's specific to neuroscience. And this is where you actually have specific information about neurons and neuron data that you might want to put into your model. So I think you can see here it's sort of self-explanatory. The model is parameterized by the name of a model file and for example, a simulator. And the test is parameterized by some sort of information about the test parameters. So for example, we're testing the Rio base, which is the amount of current necessary to evoke a single action potential in a cell. Here's some observational data that would be necessary to parameterize that test. You don't have to put in your own data. You can get data from NeuroElectro or from the Allen Institute or from Blue Brain, for example. But you can also do data from your lab, which is sort of the example case here. So you can actually use many different data sources. Many are built in, or you could bring your own. We have resources for parallel execution of tests and doing optimization of parameters for these neuron models, and built-in test classes for the kinds of things that people actually like to test. So we have over 40 distinct test types for neurons, and then several others for ion channels as well. And we also are big fans of reproducibility, so all of this and SciUnit as well. We have Docker containers that we use to be able to ship this kind of a process to other machines. So here is a use case of this approach. So C. elegans is a very small roundworm that lives in water, and there is a project called the Open Worm Project, which some of you may know about, and those who don't should go check it out. It's a project to simulate all of C. elegans, the entire organism. So this is, of course, a very multi-scale model. You're going to have ion channels in the model. So here's an example of model validation for an ion channel. This is an ion channel called EGL19. It's in a certain kind of cell in C. elegans. And if you run a particular kind of test that checks whether the IV curve, which measures the current at particular voltages, you want to see whether this particular model passes the test. So the test is parameterized by the black data, which comes from experimental literature. The model, in response to the same steps of current, produces voltage steps, produces the red curve. So you can see that's not a very good fit. We can say this is a failure. But you can look at other features too. You don't have to go to ion channels. You can step up to whole cells. And you can ask whether the action potential shapes are correct, or whether the spikes have the correct distribution, if they're bursting the right way. Or you could step all the way up to the behavior of the organism and ask, does the model organism, which is schematized here in B, does it obey the experimental, does it agree with the experimental data produced from video recordings of C. elegans swimming around in a dish, which there's many terabytes of data available. So all of that data can be put onto the website, which can be used as a progress tracker for Open Worm. So Open Worm is this big project with dozens of people from all around the world, and you want to be able to track the progress of the model. Is it getting better to explain the data, or is it getting worse? What are the deficiencies? Where are the strengths? And this is sort of a way of showing this on the website. So I'm going to dive in to the website, but before I do that, I want to show one more example. Sharon talked about the beginning of this slide with models of cosmology, right? So it was very clear that the green cases were the ones that were successful. So for example, general relativity, Einstein's model passes all the tests. The Ptolemy's model passes very few. So here are 7 examples of models of mitral cells from the literature. And here's 6 different tests taken from experimental data. And so you can see how well do these models do on these tests. And depending on which tests are important to you, you may choose one model over another. If you don't care, for example, about the dynamics of within the action potential, then you might not care about a low score on something like a width test for an action potential. But that might be something that's really important to you. If it is, you might not want to use models that get low scores in those tests. So I'm going to play this small video. This is from the website. Here you can see a bunch of test results that are stored in the database, and they're for different models like the ones I just showed in the last slide. And then just information about the score, the model type, and where it came from. And we're just going to go through here and look at the sidebar. You can look at some tests here. These are just some dummy tests that are used for this video. But for each test, you can edit that test, or you can produce a new test based on those results. Or you can create an entirely new test where you can, for example, select a test class, like that it's a test of action potential width, for example. You can go and you can do the same kinds of things with models. where you can look at all the models that are currently in the database, or you can provide your own model. So this is not a place to do modeling. This is a place, though, to register models that you want to run tests against. Here you can also copy those models as well and run and make small changes and make further tests. Here's where you would schedule the models and the tests. So in this case, you pick the test that you want to run, and you pick the models that you want to run. And you can see in the bottom below that, it's already populating the little grid. And that grid is not the results of the test. That grid is just showing you, are these the kinds of tests the model can take, right? So some models won't be able to take some tests because they aren't that kind of model. You can't look at the width of an active potential in a point process model of a neuron's firing, for example. So that's the website, and I'm not going to take you through a demo of the live website, but you can ask about it in questions. It's something that we're going to be releasing in the next year. So I want to thank all our project partners who have been working with on this to implement this. We have been using data from Neuroelectro, which I showed earlier, and also from the Allen Institute to constrain tests. OpenWorm has been a great playground for exploring all this model validation work, and they've been very supportive of it. And I think it's a great place where you have models at all different scales, many different diverse kinds of experimental data, and it's a real challenge to integrate all of that together. But I think what we've been showing is that we can actually fit all of that into the same framework. Open Source Brain has been very supportive as well. A great place to not just define, but also explore models in great detail and do collaborative modeling as well. So you can go to Open Source Brain, pick models, and you can just start working on them. And the Human Brain Project, which is using SciUnit in a number of use cases inside their project to check whether their neuron models agree with the experimental data they've collected. All of this is on GitHub. This is an example for SciUnit. We are very interested in keeping everything open source. And finally, I'd like to thank, in addition to Sharon, Russell Jarvis, who's in the lab, and Justice Bergiolis, who worked on NeuroML DB, Giovanni for hosting and for helping us develop this SciDash portal. So the great thing on MetaCell is these guys, they are scientists and neuroscientists who really understand how to work with scientists. They're not random software developers you would find somewhere. They're software developers that are their wheelhouse is absolutely working with scientists to create content. And Stephen Larson, the CEO of MetaCell, who's also been very supportive, and of course our funding agencies. That's it.
All right, thanks, Rick, and thanks for the mention. So right now, I think it's time for questions. And I saw that during the talk, the questions were already popping in. I'm gonna go back to my screen and present the Q&A. Okay, so people feel free to submit any questions you might have. We already have some, so we can start taking those. So first question from the audience, and Rick and Sharon, you just volunteer for answering. So how do you tell the tool how to read the outputs of your model? Do you need to save it in some specific format?
So it depends on the type of model. So we in this quest to sort of align ourselves first with standards, which cover a very large number of models, right? So if we use NeuroML, for example, that already gives us access to thousands of models. This becomes pretty easy. It's just the model file is what it is, and our code will handle all the rest. If you've say written a custom model, in MATLAB or something, then there's some work that you're going to have to do to expose the things that you want to test to the model. For example, let's say your model is something where you can inject current into, then there needs to be a function that you wrap with SciUnit code to do that tasks. If you're in an intermediate case, like let's say it's a neuron model made in Neuron, the Neuron software, then that's something that we can handle as well with a little bit of conversion. Sharon?
Yeah, I don't really have anything to add.
Okay, so we can take another one. Thanks, Rick. Do you only handle a certain type of models, or can you handle any model? For example, do you handle disease models?
We can handle any kind of model in principle in SciUnit. So SciUnit is completely domain agnostic. It does not require that it be a neuroscience model or a neuron model or anything. It doesn't even really require that it be simulatable. It could be a model that simply makes a certain set of predictions. And if those predictions are exposed through SciUnit methods, then that model can be tested. So there is domain-specific work to be done, right? So Neuron Unit is doing that domain-specific work for neuroscience and for neuron modeling. But there are some of our project partners have used SciUnit without Neuron Unit, right? So they've written their own things using SciUnit to test their models.
Okay. Do you guys want to take another one, Sharon? Do you want to add anything to that?
Yeah, that's good.
Okay, so we'll take another one then. Does your tool take care of running the simulations to get the output, or what if you need supercomputers to run the simulation?
Yeah, so the tool does need to be able to run the simulation if it's something that's required for the test. So you saw when Rick was giving an example from Neuron Unit that he specified the back end of Neuron in that example on one of those slides. And so, you know, it's set up now to handle a number of different backends, but adding something completely different, as Rick said, might require a little bit more work. But it's, you know, the whole setup is designed to be able to deal with that. In terms of supercomputer, you know, Rick can say, I mean, it's set up already, as he mentioned, to be able to handle parallel tests, but maybe Rick could say a little bit more about the details of that, maybe Neuroscience Gateway or, you know, whatever we've been using.
Yeah, so our graduate student Russell has done a lot of work on making this parallel testing work, and he's run, for example, Neuron Unit tests on resources with you know, dozens of cores successfully, and he's done optimization with those as well. So you optimize against a collection of tests. So you want to, let's say, find the set of parameters for a model that get pretty good scores on a whole bunch of tests instead of just one test. And so he's done that on pretty, you know, sophisticated computational resources. We've also done a Neuroscience Gateway where you can get a larger allocation, maybe hundreds of cores as well. If you're testing something that's outside of Neuron Unit, then you should talk to us to help support that for supercomputer resources. But it's definitely, we've done that for specific use cases.
Right, and also something we're working on the web application side of things is in integrating SciUnit and Neuron Unit with external servers. So right now, the stuff that's going to get released next year, we're going to go run the simulations to an external server because we need to make that multi-user. So there's no reason why we couldn't integrate a cluster of the same type. So
right,
on top of what Rick and Sharon said already.
The schedule that we showed on the web portal is definitely distinct from the machine that runs or at least from the resources that run the simulation itself.
Yeah. Okay, so another question from the audience. Are you going to the next level of creating better models after testing, or do you stop at showing what models are best?
Yeah, so I think Rick touched on that a little bit, this idea of optimization. So that's definitely something that we've been working on and that we're interested in. You know, as he pointed out, if you have the ability to do tests and evaluate how well models do on those tests, then, I mean, that's the information that you need during optimization for goodness of fit. You know, if you're trying to say, how good is my model improving during optimization?
In most cases, I'm stopping at how well does it do, because then the modeling community can then go take the next steps, right? I'm not an expert in every modeling type. In OpenWorm, for example, I consider my role to be model evaluation and not model creation. But in other cases, we are actually taking the next step and making the models better. So for example, we're making optimized reduced neuron models of every cell type in the brain. So for every cell type you can think of, for which there's data, we're trying to make reduced neuron models of different classes. you could use that agree with all as much as possible, given the model, with all the experimental data that's been reported about that neuron type, and then you could use that in a plug and play way. Let's say you're building a model of a certain brain area, and you say, okay, I need a cerebellar granule cell, I need a cerebellar Purkinje cell, and so on. You'd be able to, and you have a particular level of abstraction that you're looking at, and maybe it's not full-blown multi-compartmental models. Maybe it's Itzhakevich neurons or it's exponential integrated fire neurons or whatever. You can just go and get the one that's already optimized to match that neuron type, and then rather than having to code your own and sort of figure out whether it matches the experimental data or not.
Those aren't available yet.
That is like the last aim of grant that is in its last year. So soon.
Okay, so I think we have a few more minutes. We can take a few more. There's one question that it's interesting, it's a bit long, so I'm going to paraphrase. How do you how you define what a unit is? Because in some things that you showed, you were showing like system level tests, and then you were going down to some narrow aspects of the parameters. So how do you, the question, the core of the question is, how do you find a unit? How do you define a unit?
I would say a unit is, in this case, defined as something that gives a scalar output. So, for example, the width of an action potential is it's not a vector. It's a single value. But so is how fast does the worm swim in a certain direction. So as long as it's a single value, there could be a collection of these single values that are a suite of tests. You could group all the tests of ion channels and all the tests of spiking and all the tests into suites. And so it would be suites of individual unit tests. But for an individual unit test, there's nothing about scale in that decision. That's it's really a matter of is there an analysis you can do, and the output of that analysis is a number, maybe it has a mean and a standard error or something associated with it. And then you can then do the corresponding test on the model. So it's one thing, it's one scalar output, but it doesn't mean it can't be a broad systems level phenomenon.
Right. So it really depends on the level of abstraction that you're placing yourself on.
I've unit tests that I've written for InsideUnit for psychophysics, right? So entire perceptual or behavioral outputs of human beings, and so they're still considered unit tests.
Right. All right. So I think we have time for one or two more. There's a question about how to get specific examples of further materials for the kind of tests that are supported. So maybe you want to plug in some links, Rick, point people at the Jupyter notebooks or resources.
The main link, what's the best way to do this?
I can give you control again of the screen.
Sure. I'm going to I go, do I have control? Is it sharing the screen or not?
No, it's sharing my screen. I'm just going to give it back to you now. Okay, you have control now.
Okay, is it now showing? Oops,
yes, it's showing your screen. Yeah, it is live.
Exit the slideshow.
Yeah.
Exit. Showing something in Chrome in now?
Yeah.
Yeah.
I'm just going to exit full screen so I can go back and forth between a few tabs. But
right.
Basically, here are some go to www.SciDash.org. You get the main sort of informational page, right, which has this graphic that Sharon showed. It shows you some of the core technologies, and you click on the links, take you more about SciUnit. There can be a conference paper about it that we wrote several years ago. You can also go through some tutorials, API documentation, and of course the link on GitHub to go and see what's going on. So you can see where we stand on code coverage, and here's more information about it. Neuron Unit, same thing. That's also available from the main SciDash GitHub repository or from the website. So you can go to the Neuron Unit webpage, and you can inspect the code and see what the coverage is. You can also go to our docs, which are mostly API documentation. I wouldn't say there's a lot of high-level documentation there, but you can read about it here. You can read through these tutorials. You can see some of the previous presentations, and you can see some examples that are given in here. So all this code you see, if you're not a person who writes, if you are a person who writes code, you may love it. If you're not a person who writes code, then I think waiting for the SciDash portal to be ready might be the best approach. So there's something here for everyone.
All right, so that's great. And there's more questions, but we're out of time. So I want to thank everybody, Sharon and Rick, for coming and presenting the work, and the audience for participating in the polls, and of course in the Q&A. So yeah, I think thank everybody. Thanks everybody. Thanks Rick and Sharon. Thanks for coming, and we'll see you next time.
All right, thank you, Joe.
Thank you.
Thanks, bye.